Alright, good evening class. This is going to be the first online module, and there's going to be a total of 3, maybe 4 online modules. They should be between 8 and 8.5 minutes each. This week we're actually going to go over the central nervous system, and since I'm not here to teach you, you're going to need this online lecture in order to do well on the next exam. As we begin the chapter on the central nervous system, I want to tell you that the way that I broke down the chapter is very, very similar to what you see in the book. However, there are some non-essential items, as far as I am concerned, for you in this class that I did not include on our notes. You'll not be responsible for them for the exam, only what's in my notes and what is on the study guide. Now, as you look at the notes under the Nervous System 3, I emailed them to you and they are also present on the website. Make sure you check them out. I put in the bottom right hand corner a number. So every single page has a number, and that is from page 1 through 26. So today we'll be going through slides 2 through 26, beginning with slide number 2. Slide number 1 is the cover slide, and slide number 2 reminds you that the central nervous system is the brain and the spinal cord. Now remember, the peripheral nervous system is going to involve everything else, including cranial and spinal nerves. But the central nervous system is going to be the brain and the spinal cord along. So that's what we'll talk about tonight. On slide number 3, as you advance, one interesting thing about the nervous system, and specifically the central nervous system, is development begins very, very early on. So when we're talking about embryology, or the development of an embryo in utero, what we find is that neural tissue develops very, very early on, and specifically around the 2nd week development begins, and it develops from one of the 3 types of embryonic tissue. There are 3 types of embryonic tissue, and the one that you need to be familiar with for this module is the ectoderm. Ectoderm, E-C-T-O-D-E-R-M, ectoderm. And the ectoderm is kind of interesting because it starts out as kind of a flat sheet, but as it begins to develop, there tends to be a fold in it. And the initial segment that fold arises from is referred to as the neural plate. And the neural plate is going to begin, and it will develop a little bit dorsally on the embryo. As it continues, there tends to be a little pocket that forms in it, and your book refers to this, as many textbooks refer to it, as an invagination, and that means that there's a little bit of a crevice or a fold that develops inside of it. And this invagination, or this fold, eventually leads to the formation of what is called the neural tube. So the neural plate is laid down initially by several cells, and those cells coming from the ectoderm. So as the ectoderm begins to develop, a single strip inside of it forms the neural plate. As the neural plate begins to bend and fold, an invagination or a groove develops in it. And this leads specifically to the neural tube. Now, this neural tube contains a neural groove, and eventually there will be what is referred to as the neural crest. And the neural crest is going to form due to a complement of cells called the ganglionic cells. Now you'll remember that in the peripheral nervous system we have ganglionic cells, if you remember that from lab the other night. Ganglionic cells are going to be present inside of the periphery, but we find them at this point in time that they are the developmental cells for both the central nervous system and the peripheral nervous system. So that's kind of a little bit of a breakdown of how in the embryo the ectoderm is responsible for laying down the groundwork for the development of all of the nervous system, but specifically in this case, because we're talking about the central nervous system, the brain and the spinal cord, that is developed from the ectoderm. So as you advance your slide, now we're going to go on to slide number 4. And in slide number 4, what you see is that there is a hierarchy involved with the development of brain tissue. Now, if you'll remember when we talked about anatomy and physiology, we said that there are 3 guidelines as far as the studying of anatomy and physiology, and that there are three principles that exist, and one of those is the hierarchy that exists inside of the human body. Now, one of those places that is manifested very clearly is inside of the nervous system. As we begin to look at these hierarchical developments, one thing that you need to understand is that the way the brain develops from single cells, then it begins to become multiple cells, and of course tissue is developed. And as tissue begins to develop and arise, what we find is that there are primary features that begin to erupt and be elicited inside of the nervous system. But then after these primary features exist, secondary features are manifested. And then there are eventually tertiary and quaternary features that exist. But we'll talk about the primary features first of all. And the primary features that exist from very, very early on in development are going to be the forebrain, the midbrain, and the hindbrain. And you may remember those from class the other night as well. The forebrain is going to make up the majority of the brain itself, what we see when we see the brain. The midbrain is going to be located as a very, very deep structure. And then the hindbrain is going to be composed of parts of the brain stem leading down to the spinal cord. So kind of reorienting you and refamiliarizing you with that concept, that there is a forebrain, a midbrain component, and then the hindbrain as well. Now, the secondary features that arise from the primary features that are seen are going to be the telencephalon, the diencephalon, the metencephalon, and the myelencephalon. Now, all of these are going to make up different features whenever we start talking about tertiary and quaternary structures. So the features that we have learned in the lab and the features that we'll go over tonight and what their functionality is, all of those rise from these secondary structures. And as a matter of fact, some of them continue to carry on the name. So things like the diencephalon, that portion of the brain carries its name even into adulthood. So these names aren't just relegated to a developmental phase. They actually tend to keep their names as we get into secondary and then further more advanced structures. As you move on to slide number 5, and I don't want you to be familiar with any of the developmental aspects as far as functionality or structures involved with secondary features. I think that's just going to add a lot of complexity to what you already have to know, and this is going to be a pretty full exam as it is already.